Hi. We're back and we're still talking about chemical reactions. So we just finished talking about the oxidation and reduction reactions, which go hand in hand and are often termed redox reactions. So now let's talk about exergonic and endergonic reactions. An exergonic reaction is a reaction in which the reactants themselves, the molecules that are entering into the reaction, have more energy than the product. And so we release energy. Energy exits, exits the reactants as they become the products. Endergonic reactions are reactions where the reactants have less energy than the products that they will produce. So in an endergonic reaction, energy goes into the molecules, the products. Exergonic reactions are breakdown reactions. So they are decomposition or hydrolysis reactions. Whereas endergonic reactions are synthesis reactions where we're building something up. So if you have an exergonic reaction, you're releasing energy because you're breaking bonds. If you have an endergonic reaction, you're taking in energy because you're building bonds. Exergonic reactions can drive endergonic reactions. Here is an exergonic reaction. Here we have the reactant is glucose, and then we have oxygen. Glucose is a high energy molecule compared to carbon dioxide. So glucose is broken down to carbon dioxide and water with the help of oxygen, and energy is released. That energy that is released from this exergonic reaction can drive this endergonic reaction right here. So here we have amino acids, so like thymine and thymine. And we can build peptide bonds between the amino acids, producing a protein. So we have to have energy supplied to produce these proteins. So when we need energy, we use ATP. ATP is chemical energy of our cells, and it can be continuously formed and then broken down to give us energy. So ATP is formed when exergonic reactions occur, and they release energy. They release energy in the form of ATP. The ATP then immediately can be broken down into ADP and a phosphate to release the energy. And then it can be built back up into ATP when we don't need to use the energy. So here we see ATP, high energy molecule. The high energy bond is between the second and third phosphates. We release energy when we break ATP down through this exergonic reaction. And we then end up with ADP, adenosine diphosphate, with an extra phosphate, inorganic phosphate, just sitting there. When we need more ATP, we can take adenosine diphosphate, add the phosphate back to it to produce more ATP. So this is a cyclic reaction. Reactions occur at certain rates. A reaction rate is the measure of how fast some reaction will occur, and that is dependent on the amount of energy required for the reaction to occur. So activation energy is the energy needed to either build up or break bonds. We can use a lot of different things in labs to increase the rate of a reaction. But the most common mechanism to reduce the rate of a reaction is by adding enzymes. And an enzyme is a catalyst composed in general of proteins that will then either build up or break down different reactions. So we talked a little bit about these in chapter 2 already, so hopefully you already have a good understanding of enzymes. But enzymes decrease the amount of energy it takes for reactions to occur, and they do this by lowering the energy of activation that is needed. So in our body, just assume all reactions occur with the use of enzymes. So all reactions will occur, I will just say that. We're always leaning towards entropy, so towards less stability, more randomness, which means all reactions will eventually occur, even in our body without enzymes, but the speed of the reaction would be so much more slow that it would not allow us to survive. So let's look at how much faster an enzymatic reaction will occur. Here we have sucrose being broken down into glucose and fructose. So they're going to use an enzyme, probably sucrase, which is named for its substrate. And that enzyme decreases the energy needed for sucrose to be broken down. Without that energy, you have to wait until you have enough energy to get the reaction to occur. So it's reducing the amount of energy, the activation energy, needed to where the reaction will occur much more quick. And enzymes work really well. Not only do they work excellently, but they're not used up in a reaction. So enzymes are globular proteins. They have a specific site called an active site, and this is where the substrate will sit. The active site is specific to its substrate, so whatever its substrate is, only typically that substrate will be able to bind effectively and have the reaction occur. So if you have, say, a sucrose molecule, sucrase will break it down. But if you have a different type of molecule, like, say, lactose, sucrase can't break lactose down because they don't have the same shape. This is often termed lock and key, but it's really more of a binding and, what do I want to say? Hold on. The term induced fit is a better term for how enzymes and substrates work together. The enzyme and the substrate bind, and then the enzyme itself gives a little hug to the substrate and then catalyzes whatever reaction is going to occur. So enzymes are proteins, and they're going to be found inside of cells as well as in plasma membranes, and some enzymes are going to just be secreted into the lumens, depending on the type of enzyme we have and what the enzyme does. So this is basically how enzymes work together. The substrate binds to the active site. The enzyme then changes shape slightly, resulting in kind of a hug. This is that induced fit that I was talking about. Enzymes aren't static. They're dynamic. They can change a little bit. They can form to the substrate. This causes stresses to form, either building new bonds or breaking bonds, and then the products are released. Enzymes are never broken down or I won't say that never, but enzymes aren't used up in a reaction, so they can catalyze millions of reactions every single minute, depending on the type of enzyme. So this is showing a decomposition or a hydrolysis reaction, where you have a larger molecule, lactose, binding to lactase enzyme, and the products, glucose and galactose, are released as lactose is broken down. You can also produce larger molecules from enzymes. So in this case, we have a glycogen synthetase enzyme, which functions to synthesize glycogen from glucose. So you take glucose monomers, they bind to the active site, and when the glycogen synthetase squeezes the glucose, they build bonds between them, producing glycogen, which is the storage form of sugar or glucose in our cells. Sometimes enzymes need helpers. These helpers are called either coenzymes or cofactors. A cofactor is an inorganic molecule, such as zinc would be an example of an inorganic cofactor. Organic cofactors are coenzymes. And so the example of organic is usually vitamin B as an excellent example of a coenzyme that is often used in the help or to help enzymes and substrates work together properly. I mentioned this before, but enzymes are named for their molecules that they work on. So lactase converts lactose into glucose and galactose. Sucrase converts sucrose into glucose and fructose. Reaction rate is going to be dependent on each individual enzyme. Most enzymes can catalyze millions of reactions per minute. But if we want to increase the chemical reaction rate, we can increase the amount of enzyme, or we can increase the amount of substrate. But we can only increase the amount of substrate to the point of enzyme saturation. The term saturation in this case means that an enzyme has enough substrate to continuously produce product. And so if a substrate is, or if an enzyme is completely saturated, it's going to produce the maximum amount of product it can for every single minute, which means you can't increase the results or the product production by adding more substrate. But you can still increase the product production by adding more enzyme. So if this doesn't make sense, bring it to the class, because I can talk about it even better. In person, I can draw pictures. I'm really good at explaining how enzyme concentration can be affected by either increasing or decreasing substrate as well as enzyme itself. Okay? And so here's an example of what I was talking about. Here we have some type of enzyme, and there's the substrate. The increase in in the amount of substrate that we add will increase the amount of response until enzymes are saturated. At that point, enzymes can't work better than they already do. If they convert 1 million molecules every minute, then they're going to continuously convert 1 million molecules every minute. But they can't convert all of a sudden more than 1 million molecules, because they're already working at their optimum. So there are different factors that can affect enzyme activity. One of them is temperature. So the 3D shape of an enzyme is going to influence its function. Structure determines function. You'll never get away from this. And so its structure is dependent on the environment around it. if we get a temperature that is too high, that can actually cause enzymes to start breaking down. So a typical fever in the body is going to make enzymes work a little bit better. It's just like warming up your muscles. When you want to run at your best or exercise at your best, you first stretch and warm your muscles up. Same thing happens with enzymes in the body. If you warm up the enzymes a little bit, they're going to work a little better. But if you increase the temperature too much, you can cause proteins to break down. That's known as denaturation. And here's an example of denaturation. There's your egg. Albumin is protein. It's that clear viscous material. And as we increase the temperature, the viscous material changes in shape, becoming not ineffective, becoming unfunctional, non-functional. That's what I want to say. And so it no longer can do its job as a rubbery white structure, but it could do its job here the way it was supposed to. So all proteins in the body typically work best at body temperature, so around 37 degrees Celsius. But they can work well up to 40 degrees Celsius really effectively. And they can even work at a little bit lower of a temperature. But if we increase the temperature too much, that can cause denaturation of the proteins. If we decrease the temperature, enzymes become very rigid and they no longer function as effectively. They become very, very hard to work with. So remember, enzymes aren't lock and key. They have that induced fit, so they kind of hug their substrate. If they become too rigid, they can't hug their substrate. pH can also influence enzyme activity, and in the body, we have multiple pHs. So depending on the region where the enzyme functions, it will have a different optimal pH. If we increase or decrease the pH too much, it can cause the enzyme to denature again. So this is where I like to look at different proteins. So if you have a pH that's optimally, or no, not optimal, I'm sorry. If you have an enzyme that has a neutral pH as its optimum pH, then anything too acidic or too basic is going to cause damage to that enzyme. It will denature. But there are some enzymes that work best in a pH that's very, very low. So an example, pepsin is an enzyme that breaks down proteins in the stomach. That enzyme works best at a pH of 1 or 2. Inhibitors can influence enzymes. Inhibitors are molecules that will either bind to the active site or bind to another binding site called an allosteric region on the enzyme. So competitive inhibition is the binding of a substrate to the active site. They're competing for that binding site, and typically inhibitors that competitively inhibit molecules binding are poisons, and once they bind, they don't unbind as easily. The best example of this is carbon monoxide binding to your hemoglobin. It won't unbind unless we force it to unbind. Non-competitive inhibition is where you have a molecule that binds to a different region on the enzyme. So another binding site, typically called the allosteric site. And so non-competitive inhibition occurs when an allosteric inhibitor binds, changing the shape of the substrate. And that can influence how effective that enzyme is. So allosteric inhibition is often used to maintain amounts of different molecules in the body. If we don't need something, then we will inhibit that protein from producing more product by having an allosteric inhibitor bind, and then the substrate will no longer be able to bind. We can add or take away phosphates, and that can also influence enzyme activity. So if we have an enzyme that is in our system that typically isn't turned on, we can turn it on by either adding a phosphate or by taking something away from it. The best example for this is pepsin, again, which is found in our stomach. It's produced by our stomach cells. And when pepsin is released, it enters so it's released as a non-functional protein, and it's known as pepsinogen. And we have to cleave a portion of the pepsinogen to allow it to become functional pepsin that can break down proteins. I'm going to stop here because now we're going to get into aerobic respiration or cell respiration, which we will focus on for the majority of the rest of this chapter. So yay on that one, right? Bye.